Good afternoon, everyone. So, next chapter that we are going to start in the cardiovascular pharmacology, it is diuretics. So, as we have studied about diuretics in the chapters, that is drugs used in case of hypertension and some other diseases also, we have introduced you to the point that is diuretics, and also we have seen that what function these diuretics play in those particular diseases. So we will study in detail about diuretics in this particular chapter, and we will be studying about the pharmacology of the important drugs. Okay, so first of all, before we actually move to the pharmacology of the diuretics, we should learn about, or we should revise about the renal physiology, how urine forms, what are the different parts of the nephron, then what is the structural specificities of these particular parts, that all we are going to cover. And then we will actually start with the classification of the drugs, and then we will study pharmacology of each and every class. Okay, so first of all, renal physiology. So basic urine forming unit of the kidney, it is nephron. So the parts of nephron, we have two parts, that is, the first part is the renal or Malpighian capsule corpuscle, which consists of a capsule, that is Bowman's capsule, and we have a tuft of capillaries, which is nothing but called as the glomerulus. Okay, so then we, second thing that we have, it is tubular portion, which starts from the PCT stands for proximal convoluted tubule. Then next comes, that is the loop of Henle, which has three main parts, that is uh, descending thin limb, then this is ascending thin limb, and this TAL stands for thick ascending limb. Okay, so both in this case, that is DTL and ATL, T stands for the thin, okay, L stands for limb, and D stands for descending. Descending means it descends into the medulla. Okay, and ascending, it is now rising up from the medulla and moving towards the cortex part of the kidney. Then next to the loop of Henle, we have the DCT. DCT is distal convoluted tubule, and lastly we have the collecting duct. Okay, so the processes which are involved in formation of urine, they include, the first one is the glomerular filtration. So the Bowman's capsule and the glomerulus, it takes part in this particular process, that is glomerular filtration. And you have already studied the structure of the filtration membrane, so that part, it is like now we are revising the things. Okay, so next we have the tubular reabsorption, in which the essential electrolytes, they are reabsorbed from the tubular fluid. Okay, so mainly we are concerned with the absorption of sodium over here, sodium and the water, in case of the diuretics. Okay, then we have tubular secretion, the third process, in which the waste products, they are secreted into the tubules. Okay, so next is structural specificities of the tubule. So when we are talking about the PCT, okay, so we are not right now talking about the glomerulus and the Bowman's capsule. We are talking about the tubular part of the nephron. So the first part is the PCT, which is in continuation with that of the Bowman's capsule. So what about PCT? There is about 65% of the sodium which gets into the tubular fluid, it is absorbed in the PCT. And also the PCT, the membrane, it is water permeable. Okay, then we have a descending thin limb, so it is highly permeable to water, but it is less permeable to urea and the sodium. Okay, so what about ascending thin limb? It is permeable to sodium and urea, but it is impermeable to water. understand the permeability to the water, it uh, it differs between the DTL and ATL, and this permeability of these particular regions to the water, it is helpful, or it is causing that counter current exchanger, one of the phenomenon that you have studied in human anatomy and physiology. Okay, so if you don't remember it, I would suggest that you should go through this particular chapter from your book, that is Tortora. Okay, so that if you read that particular point, it will be easier for you to understand the pharmacology of diuretics, because the pharmacology of diuretics, it solely depends on the anatomy and physiology of the urinary system. So those concepts you need to be cover up, or you need to learn very diligently to understand the concept of pharmacology of diuretics. Then we have next point, that is TAL and the DCT. So this particular part, it is permeable to sodium chloride and it is impermeable to water. So what happens over here is that this is the PCT, then we have loop of Henle. So this is descending part of the loop of Henle. So this is a descending thin limb. Then we have ascending thin limb, and we have the thick limb. Okay, so that is the TAL process. Okay, then we have the uh, DCT part over here. Okay, so this is TAL, this is ascending thin limb, this is descending thin limb, and this is PCT. Okay, so when the fluid is moving through the PCT, as 65% of sodium is absorbed, so this also the water permeability is good over here. So the whatever fluid we have over here in this PCT, it is isotonic with that of the extracellular fluid or this interstitial fluid. Okay, so as this fluid goes downwards to this hairpin turn. Okay, so the permeability to the water is there, but the this particular portion, it is impermeable to urea and sodium. So what happens, the tonicity of the fluid which is there inside this tubule, it increases. So at this level, the fluid inside the tubule, it is hypertonic. Okay, so when this fluid moves into the ATL portion, what happens is that this portion is permeable to the sodium and urea, but it is impermeable to water. So the hypertonicity of the solution which was there over here, it is now becoming isotonic. Okay, so tonicity is decreasing. Why? Because the sodium which was retained in the fluid till now, it is now moving out. Okay, so what about this TAL portion and the DCT portion? These two portions, they are permeable to the sodium chloride, but they are impermeable to water. So what happens in this case, in both of these tubules, the sodium and chloride is moving outside, but the water is staying inside. So because of it, the tonicity of the fluid which is there in this tubule, it is decreasing. Though, so the whatever fluid we had over here, it is hypertonic. When it reaches to this particular point, it becomes hypotonic in nature. Okay, so at this particular part, we have another set of cells that are called as, or that particular apparatus is known as juxtaglomerular apparatus. And this macula densa cells of it, they actually sense the amount of sodium which is present over here. Okay, so if the amount of sodium which is present over here, it is more as the normal limits, then they release or they cause decrease in the GFR. Okay, and this decrease in the GFR, it is carried out by release of the prostaglandins by these macular densa cells. Okay, so the fluid then moves into the DCT as the sodium and chloride absorption reabsorption is continuing. The tonicity of the fluid further decreases. Okay, so that's why these particular segments of the nephron, that is TAL and the DCT, they are called as the dilating segments of the nephron. Okay, so the next part or the last part of the nephron is the collecting duct, and the water permeability of this collecting duct, it depends on the presence or absence of the ADH. ADH is a type of pituitary hormone which is released in a response to the body condition. Okay, so if body condition is dehydration, then in that case the body needs to retain whatever water it has, so the ADH will be released in that case. Okay, so this ADH will cause, as the name suggests, it is an anti-diuretic hormone. Okay, so it will prohibit the excessive excretion of the water through the urine, so it will cause retention of the urine. In presence of ADH, there will be expression of the water channels into the collecting duct, and through which the water will move into the interstitial fluid. But when the ADH is not there, the collecting duct portion of the nephron will be impermeable to the movement of the water. Okay, so next is principles of diuretic action. So how diuretics they function. Okay, so this is the principle. So diuretics are the drugs that increase the rate of urine flow, and they are clinically useful diuretics. They also increase rate of sodium excretion, which is called as natriuresis, and often accompanying anion, usually the chloride. Okay, so the most clinical applications of the diuretics, they are directed towards reducing extracellular fluid volume by decreasing total body sodium chloride content. Okay, so although continued diuretic administration causes a sustained net deficit in total body sodium, the time course of natriuresis is finite because renal compensatory mechanisms, they bring the sodium excretion in line with the sodium intake, and this phenomenon is known as the diuretic breaking. Okay, so what we have just said is that usually what happens is that when we start with the diuretics, what happens, the sodium excretion increases. Okay, so this sodium excretion, it is not like continued process. It will, to a certain extent, the sodium excretion is allowed, and when that certain extent is reached, the body starts taking, or body takes some compensatory mechanisms, it activates some compensatory mechanisms which function to increase in intake of the sodium. So whatever loss of sodium that has occurred because of the diuretic intake, that is being matched by the uh, increase in the intake of the sodium. Okay, so this is nothing but called as diuretic breaking. So these compensatory mechanisms, they include activation of the sympathetic nervous system, then activation of the renin angiotensin aldosterone axis. Then next mechanism is decrease arterial blood pressure, which reduces pressure natriuresis. Then we have renal epithelial cell hypertrophy. Then we have increased renal epithelial transporter expression, and perhaps alterations in the natriuretic hormones such as ANP. So all of these compensatory mechanisms may be all together, they will start and they will compensate the loss of sodium that has been caused due to consumption of diuretics, or any one of these mechanisms it will have dominant action. Okay, so diuretics may modify renal handling of other cations also. Example is the potassium, hydrogen, calcium, and the magnesium. Then anions, we have chloride, then bicarbonates, then the phosphates, and the uric acid. So in addition, the diuretics they also alter the renal hemodynamics, and this action of the diuretics on the renal hemodynamics, it is indirect action. Okay, so sorry for that. So next is classification of diuretics. We have main classes, that is the first one is loop diuretics or high ceiling diuretics, which are nothing but the sodium potassium 2 chloride co-transport inhibitors. I'll just mark up this so that you will understand. It is sodium potassium 2 chloride co-transport inhibitors. Examples we have ethacrinic acid, then furosemide, then bumetanide, and torsemide. Then we have second category of diuretics, that is the thiazide diuretics, which are medium efficacy diuretics and which act as sodium chloride sympathy inhibitors. Examples of it, that is the chlorothiazide, hydrochlorothiazide, chlorothalidone, indapamide, and metolazone. Okay, then we have third type of diuretics, that is weak or adjunctive diuretics. So these diuretics, example, there are three subclasses. The first one is the carbonic anhydrase inhibitor. The example is acetazolamide. Then second one is the osmotic diuretic. Example is the mannitol. And the third one is the potassium sparing diuretics, which again have two subtypes. That is, the first one is the aldosterone antagonist. Example is spironolactone and the eplerenone. Then we have the second subtype, that is renal epithelial sodium channel inhibitors. Example is amyloride and triamterene. Okay, so this particular diagram shows the, the mechanism of action or the site of action of various drugs which we have just mentioned. Okay, so we have PCT. Let's start with the first site, that is PCT, where the medication, which medication act at this place, that is the first one, that is carbonic anhydrase inhibitor. So they produce a very weak diuretic action. Then we have osmotic diuretics. They are also rarely used for the diuresis, and uh, they have, or their site of action is this, that is PCT. Then we have second site, that is descending loop of Henle. So that is the DTL, descending thin limb. Okay, so the medications which act at this particular place, it is osmotic diuretics. Also, these are rarely used for diuresis purpose. Then we have ascending loop of Henle. Okay, so this over here, which drugs act, that is the loop diuretics, which are the strongest among all the diuretics. Okay, and they are the sodium potassium 2 chloride transporter inhibitors. Okay, then we have the fourth site, that is distal convoluted tubule, and the drugs which act at this particular site are the thiazides. And also they produce like weak diuretic action or medium efficacy they have. Okay, so in the last part, that is collecting tubule and the duct, the medications which act at this particular site include the aldosterone antagonist. Why over here? Because the receptors for the aldosterone, they are present only in the collecting tubules. Okay, so they are weak to moderate diuretics, which depends on the dose. Okay, then we have second type of diuretics that act at collecting ducts, that is epithelial sodium channel antagonist. So these diuretics, they have weakest diuretic action. Okay, then we have next category of diuretics, that is vasopressin 2 receptor agonist. Okay, so these agents, they produce moderate aquaphoresis action. Moderate aquaphoresis action means what? See, these are vasopressin. Vasopressin, the ADH is also called as vasopressin. Okay, so whatever action ADH has, same action these agonist will have onto this particular part of the nephron tubule. Okay, aquaphoresis means what? When they act on it, they cause the movement of the water from the tubule into the interstitial spaces. Okay, so remember this particular chart. You will never have the problem of remembering the site of action of various diagrams.